everybody, and we are back with Unit 3, which is about heredity. We're going to start off focusing on traditional Mendelian genetics in Concept 1. So just to quickly review some things about genetics, with the exception of gametes, which we know are eggs and sperm cells or sex cells, all of our cells in our body are diploid, and that means that they contain 2 copies of each chromosome. One copy comes from mom from the egg, and one copy comes from dad in the sperm. And so you need to remember how many total chromosomes make up who you are as a human being. So the answer to that would be 46, right? 23 comes from mom and 23 chromosomes come from dad. Also, let's review some of the DNA vocabulary. So we have a gene, which is a section of DNA that provides the instructions for making a protein. We talked about these when we talked about transcription and translation. So since a gene is just a piece of a chromosome and we have 2 of every chromosome, we also have 2 copies of instructions to make every single protein in our bodies. Then you have the alleles, which are different versions of the same gene. So we inherit a full set of chromosomes from each one of our parents, but we do not have to inherit the same version of every gene. For example, mom gives you a gene for blue eyes and dad gives you a gene for brown eyes. Both of those are for the same trait, the same gene of eye color. However, it's a different option, either blue eyes or brown eyes. And then homologous chromosomes. So that's where you have the chromosomes that come from your mom and your dad, but they're matching. And what that means by they're matching is that they contain the same genes in the same locations. They can have different alleles, but it's still the same gene. So going back to that last example about the eye color is the same gene, the same trait, but you have different alleles or different options for them. So an easy way to remember this is, the prefix homo means same, so these chromosomes are the same in the fact that they contain the same genes. We're talking about Mendelian genetics, and Mendelian genetics get their name from an Austrian monk called Gregor Mendel. So he is known as the father of genetics because he figured out 3 laws of inheritance utilizing pea plants. So like peas in a pod, those type of pea plants. And so he designed and determined these 3 laws of inheritance: the law of dominance, the law of segregation, and the law of independent assortment. So just going through the basis of his experiment. So he crossed pea plants, which means that he fertilized one pea plant for another pea plant in order to create like baby pea plants. So the first thing he made sure is that he had control over the breeding. So he used pea plants that self-pollinate, which means that they create their own pollen that will pollinate themselves. So you don't have different plants mixing their genetics together. Secondly, he only used purebred plants. So what that means is that a purebred is just a type of organism whose ancestors are genetically uniform. So that means that they're going to have the same alleles for every trait that they have. So like both of their alleles are going to be for blue eyes, or both are going to be for brown eyes. They're not going to have one of each. And then he observed either or traits. So that means that they had one option or another. There wasn't more than 2 options for these particular traits. So for example, he talked about pea shape, pea color, pod shape, pod color, plant height, and flower color, because those were all only 2 option traits. So he did crosses, and so we have to understand what is a cross. So a cross is just where you have a mating of 2 organisms. If you hear something referred to as the P generation, that's just the original parental generation. The F1 generation is that first cross between the 2 original parents. And then the F2 generation is the second cross between 2 F1s. You might hear this terminology whenever you maybe get into breeding of animals. If you've ever known anybody who breeds, especially specifically like crossed dogs, like doodles, golden doodles or anything. They'll talk about this is an F1, this is an F2 doodle, and that's what that's talking about. So when we're talking about alleles, we're going to be moving forward with genetics, talking about those alleles. And we are going to assign letters to those alleles. So if the allele is dominant, we are going to use an uppercase letter to represent that. If it is recessive, we are going to use a lowercase letter to represent that. And remember that each individual has two alleles. One comes from each parent for each gene. So for every gene that you have two alleles, the one that came from mom and the one that came from dad. So that means that you are going to have two letters that can represent your alleles for a trait. You can either be homozygous for that trait, so remember homo means same, so this means you have two of the same alleles. Either both dominant, so two capital letters, or both recessive, two lowercase letters. We can also refer to these as homozygous dominant or homozygous recessive. And then your other option is to be heterozygous. So if you remember, hetero means different. So this means I have two different alleles, and the only option that I have is having one dominant and one recessive allele to have two different alleles. So this leads to the law of dominance that Mendel came up with. And what that says is that a dominant allele will express itself over a recessive allele. So dominant means that if it's present, that allele will always have the trait expressed, which means that that is seen. So for example, if brown hair is dominant over blonde hair, the lowercase b represents the allele for blonde hair and uppercase B represents for brown hair, because I know that uppercase letters are dominant alleles and lowercase letters are recessive alleles. So that means that if you inherited a dominant brown hair allele from either one parent or both of your parents, you're going to have brown hair. So regardless of whether both of your alleles are brown or whether one is brown. Remember, because it is dominant, so it's going to kind of overshadow the recessive allele even if you have a recessive allele. So recessive alleles, you're only going to have the trait expressed or seen when the dominant allele is not present. So what that means is the only way to get blonde hair is to inherit the lowercase recessive B blonde allele from both parents. So your genotype would have to be lowercase b, and that's the only way for you to get blonde hair. Because if you had a dominant brown hair allele, that would overshadow the recessive. So when I use the term genotype versus phenotype, genotype are the letters that are representing the alleles that you are inheriting. So that's going to be the genes that code for flower colors, such as uppercase F, uppercase F, uppercase F, lowercase f, or lowercase f, lowercase f. So it is representing the alleles. It's talking about the actual genes that you are inheriting. Whereas a phenotype, that's the physical trait or the physical characteristic that is seen in an organism because you have those alleles. So for example, purple flowers, blonde hair, blue eyes, all of those are physical traits. They're not the alleles themselves. So moving on to the second law, which was the law of segregation. So we know segregation means to separate, so keep that in mind as we're going through this idea here. So when chromosomes separate in meiosis, each gamete, either the egg or the sperm, will receive only one chromosome from each pair. Remember, we are taking diploid cells and making haploid cells, so I'm only going to get one chromosome out of each homologous pair. Working through this example, if a man has alleles for brown hair and blonde hair, he is heterozygous for hair color, big B, little b. His sperm cells can contain the allele for brown hair or the allele for blonde hair, because those alleles get segregated or separated during meiosis. So each sperm cell will only get one of those alleles. And then whenever it fuses with the egg, that's where you get your other to create a genetic combination that's unique to you. And then that leads us to the last law, which is the law of independent assortment. So this means that the assortment of chromosomes for each one trait doesn't affect the assortment of chromosomes for another trait. This means that you can have any combination of maternal and paternal chromosomes to be passed on because homologous chromosomes lined up randomly during metaphase. So what that means is that you can have infinite number of combinations of your parents' chromosomes because when the homologous pairs line up during meiosis 1, during metaphase 1, they don't always have to line up, all of your mom's chromosomes on the left and all of your dad's chromosomes on the right. They can mix and match as long as they're with their pair. So that leads us into kind of making predictions about what offspring or what the probability of offspring would look like based on the alleles that you have. And we can do this through Punnett squares. So Punnett squares are diagrams that show the probability of inheriting traits from parents with certain genes. We'll start off with monohybrid crosses. So the prefix mono means one. So this is when you're having a cross between two different organisms, but you're only looking at specifically one trait. So eye color is one trait that we could be looking at. The picture here is an example of a filled out Punnett square. We're going to work through how to do these, but the 4 options in the middle are the possible genotypes of your offspring. So you have the different options of what the kids could have. So let's talk about how to work through a monohybrid cross. Whenever you have these, a lot of times they're going to be word problems. They can either be complex word problems or non-complex word problems. These ones are pretty straightforward as long as you have your vocabulary down. So in pea plants, round seeds are dominant. So that lets me know I need to use a capital letter in order to designate round seeds. So I'm just going to use the capital letter A. And wrinkle seeds. So that means that wrinkled seeds are recessive if round seeds are dominant. So wrinkled seeds would be recessive. Cross a homozygous recessive plant. So homo means same, recessive means 2 lower cases. So the first pea plant is that. With a homozygous dominant plant. So homo means same, dominant means capital alleles, so that's going to be my second plant. Okay, so in this case, I'm going to have the homozygous dominant plant and the homozygous recessive plant. It doesn't matter which side you put which parent on, it's going to work out the same. So you're just going to drag and drop. So you're going to take these and go down and fill out the squares. And then you're going to take these and move these over. So this means that 4 out of 4, or 100% of all of the offspring are going to be big A, little a. And I know that that means that the dominant trait is going to be the one that's shown, which in this case is going to be round. So this is the genotype, right, because this is the alleles, and then round would be my phenotype. Okay, so did that one. Let's go to the second one. All right. Cross 2 heterozygous plants. So hetero means different. That means that they have 1 dominant and 1 recessive allele. Like that. And it says that both plants are that way. So I'll fill out my Punnett square, like so. And that gives me this. So I have 1 out of 4 squares, or 25%, is capital A, homozygous dominant. I have 2 squares, 2 out of 4, or half, or 50%, however you want to write it, is heterozygous, big A, little a. And then 1 out of 4, or 25%, is little a, little a. So both of these, since they have the dominant allele, are going to code for the dominant trait of being round. So 3/4 would be round, or 75% of these would be round. And then the only way to be wrinkled is to be all recessive. So 1/4 or 25% would be wrinkled. So remember, if it's asking for the genotype, this is what you're giving me. If it's asking for the phenotype, this is what you're giving me, because phenotype is the physical trait. So in this case, the physical trait is specifically the what the pea plants look like, what the pea pods look like. And your genotype is what are the alleles that are coding for that particular pea plant. All right, and then our last one. Go ahead and pause it and see if you can do it on your own, and then come back and watch me. So it says cross a heterozygous plant. Heterozygous is big A, little a, with a plant that has wrinkled seeds. What do I know about wrinkled seeds? They're recessive, and the only way to have a recessive trait is to have all recessive alleles. So that means that my Punnett square should look like this. Remember, if you put this parent on that side, it's going to work out the same way, so don't worry about it. So then I fill out my Punnett square, and I get this. So that means that you should have gotten 1/2 would be big A, little a, which means that 1/2 are going to have round seeds, or 50%. And then 1/2 are little a, meaning that they're going to have wrinkled seeds. And once again, if you want to do the percentage, unless you're asked specifically to not do a percentage, that is completely fine. So moving on, you have also dihybrid crosses. So the prefix di means 2. So where monohybrid, we were looking at what's the likelihood of one trait happening. With dihybrids, we're going to see what is the likelihood of having 2 traits happen at the same time. So these are a bit more complicated than monohybrid crosses, but we will work through a few of these as well. So our first example here, it asks us to cross 2 tomato plants that are both heterozygous for being tall and heterozygous for being red. And it tells me a note that the recessive traits in tomatoes are short and yellow. Okay, so being short, as a pea plant would mean that I have to have little t, and being yellow would be little r. And I know that by looking at my background information. So my step 1 is to write the genotypes of the parents, mom and dad. So it says that both mom and dad are heterozygous for being tall, so big T, little t, and heterozygous for being red, big R, little r. So then the next thing I need to do is sort the alleles. So I'm asking myself what combinations could go into the gametes during meiosis. In order to do that, I will FOIL these options. So if you remember back to math class, I know it's not math, but the easiest way to go about it. To FOIL, F stands for first. So you take the first letter out of each one of the different options. So capital T, capital R. That's the first one in each one of my sets of parentheses. O is the outer. So that's like the sandwich bread to my sandwich. So capital T and lowercase r. Inner obviously like the peanut butter and jelly to my sandwich. So little t, capital R, and then last is the last one in each one of the sets. So lowercase t and lowercase r. Step 3, write the combinations on the outside of the Punnett square. So I write my F option, my O option, my I option, and my L option. In this case, both of the parents were the same, so I can just FOIL once because they're both going to give me the same options. If they were not the same, then I would need to go back and I would need to FOIL the second parent and then put their options on the other side. So I fill this out just like I did the monohybrids, except for instead of having 2 letters in each box, I'm going to have 4 letters in each box. A lot of times I like to go about doing this 1 letter at a time. So I'm going to go through and do the T's first and then go back and fill in the R's. That just helps me keep track of what I'm doing. Be very careful when you're doing this to try to be as neat as possible, because if you're rushing, a capital T can look an awful lot like a lowercase t. So be very mindful about like what letters you choose to write with. I try to avoid writing letters where the capital letter is just a bigger version of the lowercase letter, so like O or S. I try to use letters where the capital letter looks significantly different than the lowercase letter, like A or B. These do take a little bit of time to fill out, but once you get the hang of it, it's just more tedious than anything, than really difficult. Okay, so I have this filled out now, and then I need to determine the phenotypic ratio. So a lot of times with the hybrids, they're not even gonna bother asking you about genotypes because there's so many different options. But we can look at phenotype options. So I need to come up with my 4 different combinations. So I can be tall in red. I can be tall in yellow. Or I can be short in red. Or I can be short in yellow. Those are my 4 different options based on my 2 things that were told to me in my original problem. So what I need to do is I need to count how many boxes would fall under each one of these kind of overarching things. The way that I like to do this is I like to pick out the obvious ones first, and to me the most obvious one is going to be the one that has both of the recessive traits. So I know that being short is lowercase t, and I know that being yellow is lowercase r, lowercase r. So to be short and yellow, I'm going to have all 4 recessive alleles, so all 4 lowercase letters. And whenever I'm looking at my boxes, only this one has that. So 1 out of 16 boxes are going to be short and yellow. So I can go ahead and mark that one out and get that out of the way. That way I don't look at that one anymore. Okay, so the next one that I'm going to work on, I'm going to do another one that has a recessive trait because that's easier for my eyes to find. So let's do this one, short. I have to have 2 lowercase t's, but to be red, I have to have an uppercase R and either an uppercase R or a lowercase r. Remember, with the law of dominance, it doesn't matter what that second one is. So I'm looking for two lowercase t's with a capital R. So this box fits that bill, this one, and this one. So that gives me 3 out of 16 boxes are going to be that. Okay, moving on. I've got tall in red and tall in yellow. So yellow is a recessive trait. That's going to be easier for me to find, so I'm going to do that one. To be tall, that's a recessive, or that's a dominant trait. So I need a capital T and then 2 lowercase r's. So I'm going to find those. 1, 2, 3. So 3 out of 16 boxes are going to be tall in yellow. Get that out of the way. And then finally, tall in red, I know I'm going to have a capital T because tall is dominant and a capital R because red is dominant. So 1, 2, 3, 4, 5, 6, 7, 8, 9. 9 out of 16. So for this one, what I would do is I would pause it, and then I would come back and see if your answers match mine. Okay, so in peas, purple flowers are dominant to white. So this doesn't give me a letter to use. I'm going to use A because that's easy. And tall plants are dominant to short. I'm going to use B because once again it's easy. If you used a different letter, that's perfectly fine. So a white and short plant. So I'm going to make that mom just because it's just parent number 1. To be white, that is little a. And to be short, that's also a recessive trait. So little b, is crossed with a plant that is heterozygous for both traits. So heterozygous for being purple and heterozygous for being tall. Awesome. Step 1 done. Sort the alleles. So I need to FOIL both parents. Here's parent 1, and here is parent 2. Okay. So when I FOIL parent 1, what happens is I have 4 of the same options. And then whenever I FOIL parent B, the second parent, I have these different options. So a time-saving hack, kind of with dihybrid crosses, is you only have to do the ones that are different. So in this case, all 4 of these are different, so I need to include all 4 of these on this parent side. But since this parent, all 4 of them are the same, it's just going to be really repetitive whenever I do this. So I can just write it once and kind of skip those. So then I fill these out. And if you didn't know that and you just went ahead and did all of them, you're still going to get the same fractions or percentages as I do when everything comes out in the wash. Okay. So determine the phenotypic ratio. I had purple and tall. I had purple and short. I had white and tall. And then I had white and short. Okay, so same thing I did last time. I'm going to start off with white and short. Because this one's going to be the easiest one to identify first. It's going to be all recessive. So 1 out of 4. If you did the whole thing, you would have gotten 4 out of 16, which when you simplify 4 out of 16, it's going to be 1/4. So same thing. All right, white and tall. 1/4. Purple and short. 1/4. And then purple and tall. 1/4. So they all here will have a 25% chance or a 1/4 chance of being that option. So I also can use Punnett squares to determine probability. So probability can be used to determine the possibility of getting a specific genotype. This is really helpful whenever I'm looking at more than 2 genes at a time. I don't want to do, a 32 square Punnett square. That's just too much. So what I can do is I perform monohybrid crosses for each individual gene and determine the odds of getting the specific genotype that I'm looking for in a fraction form. So 1/4, 1/2, 0 out of 4, 4 out of 4. And then I multiply the odds together, determine the likelihood of getting this combination. So let's work through an example of that. So I've got Erica here and she has this genotype. So I've got, I have 3 different genes here. I have gene A, I have gene B, and gene C. Carlos has this genotype for these 3 genes. So I'm trying to determine the probability of Erica and Carlos having a child who is heterozygous for all 3. So what I need to do is I need to create a Punnett square for gene A, as well as gene B and gene C. Okay, so Erica is big A, Carlos is big A, little a. So I fill out this monohybrid Punnett square. And I have my different options for Erica and Carlos's kids. And so I'm looking specifically for what they're asking for, which is big A, little a. So 1, 2, so 2 out of 4, which simplifies to 1/2. Okay, so then I do the same thing for trait B. Both of them are heterozygous. Both Erica and Carlos are heterozygous for trait B. So then I fill out my Punnett square, like so. And 1, 2, 2 out of 4, or 1/2, are heterozygous. And then finally, trait C, Erica is homozygous recessive, little c, and Carlos is heterozygous, big C, little c. So once again, be careful whenever you're given letters that are the same capital as they are lowercase. Make them just ridiculously different in size so you don't get confused. Okay, so then once again, 1, 2, 2 out of 4, or 1/2 is big C, little c. So 1/2 times 1/2 times 1/2. If you don't know how to do this, you multiply all the denominators together to get your denominator and multiply all the numerators together to get the numerator. So 1 times 1 times 1 is 1. 2 times 2 is 4, and then 4 times 2 is 8. So in this case, there is a 1/8 chance of Erica and Carlos having a kid with these specific genetic combinations for their alleles.